going to be talking about something a bit more specific, which is a major component of my lab's research at Tulane University right now. And so I'm going to be talking to you about the genetic basis of local adaptation and phenotypic plasticity in Mimulus laciniatus, which is a montane monkey flower. So that part is true. So when we think about how organisms respond to environmental heterogeneity, there's really sort of two main categories that people in my field of evolutionary genetics study, and that's sort of a population level genetic differentiation or phenotypic plasticity. And what we really would like to know as evolutionary biologists is, is this plastic and or population level differentiation adaptive? So I address these questions using the Mimulus laciniatus system, and this is a system I developed as a PhD student and now work on in my lab along with my students. And it is a member of the Mimulus guttatus species complex, which is a sort of emerging ecological genomic system. And it has a unique lobed leaf shape and occurs from 3,000 to 12,000 feet in the central and southern Sierra Nevada mountains. And it occurs in these it specializes in these granitic or other rocky outcrops, so pretty harsh plant habitats. So I'm going to be talking to you about leaf shape because leaf shape has been proposed to be adaptive for several different reasons. And specifically, I'm going to be talking about lobed leaves today. So lobed leaves have been proposed to be involved in heat and drought tolerance, in freeze tolerance, and also in herbivore or pathogen avoidance for plants. And today I'm going to concentrate on freeze tolerance, and I'm going to tell you about how leaf shape, variation in the leaf margin, can affect the leaf boundary layer, and how that in turn can affect freeze tolerance or other physiological properties. So the boundary layer, if you all remember from introductory physics, is a region of immobile air directly adjacent to an object's surface. So in this case, directly adjacent to the leaf surface. And round leaves have a thicker boundary layer than lobed leaves, which have a thinner boundary layer. And at night, leaves radiate heat to the cold sky if there's no cloud cover, right? So space is very cold, it sucks up all the leaves' heat. And so this can cause leaves to drop below air temperature and freeze even when air temperature is above freezing. But having a thinner boundary layer with a lobed leaf, it can actually allow leaf temperature to track air temperature more closely, because basically heat can move more easily between objects with a thinner boundary layer and the surrounding air. So lobed leaves should have increased freeze tolerance compared to round leaves of the same area. And in fact, I have some preliminary data where I did this leaf shape reciprocal transplant. I planted lobed leaf species, Mimulus laciniatus and Mimulus guttatus, which has round leaves, in granite and non-granite habitats. And I found that in the native granitic habitat, Mimulus laciniatus's lobed leaves stayed warmer throughout a 24-hour period than the round leaves of Mimulus guttatus. So we see that leaf shape can have an effect on temperature in the field. I've also done a much larger reciprocal transplant with thousands of hybrids between these round-leaved and lobed-leaved species, laciniatus and guttatus, and looked at natural selection in the field, phenotypic selection on leaf shape, and found that leaf shape, lobed leaves are under positive directional selection in granitic outcrops, so in the Mimulus laciniatus's native habitat. So therefore we can say that this lobed leaf shape is adaptive in its habitat. But in addition to being, there's a lot of variation in leaf shape across species in this Mimulus guttatus species complex, but there's also geographic variation, population level variation in leaf shape within the species range of Mimulus laciniatus. You can just see a small sample of that variation here, which is pretty dramatic. And this is when grown in a common environment. We also see from some preliminary data I gathered that there's phenotypic plasticity in leaf shape. Okay, just making sure that my computer is giving me weird notifications about the presenting mode, but if you all can't see the presentation anymore, please let me know.
Yeah, Katie, okay. That's small.
Yeah, I know, it won't go into large mode. Yeah, I already said that. You missed it. So Yeah, so
Katie, would you mind stop presenting and then re-presenting?
oh, sure. Okay, that's what I was getting weird notifications about.
Yeah, we're seeing your face instead of the presentation.
Okay. Zoom.
There we go. We're good.
Okay, cool. Okay, so there's also phenotypic plasticity in the same trait, leaf shape. So when I've grown genotypic replicates in long and short day lengths, for example, in the lab, you see that at long days there's much more lobing in the same genotype than in short days. And you can see that in this figure. The top two panels are different genotypes grown in short days, and then the bottom two panels, D and E, are those same genotypes grown in long days. So there's quite a bit of phenotypic plasticity in leaf shape, as well as population level variation. So in order to look at the genetic and genomic basis of this local adaptation or geographic variation and phenotypic plasticity in leaf shape, I've collected populations from across the species range, so over 20 populations total, sort of grouped into 3 geographically separate altitudinal transects. And then I've collected 10 to 60 genotypes per population. So we have a lot of genotypic variation across the entire range of the species to look at these traits in. I grew 10 to 16 inbred lines from 20 populations across the species range in a common environment. And I measured leaf lobing on 220 genotypes using this convex hull method that I'll tell you about in a second. And I asked, does lobing increase with either elevation or latitude across the species range? So just really quickly, the quantitative leaf shape metric I use, called the convex hull metric, I developed this during my PhD, and I measure the true leaf area. And then I measure the convex hull shape area, which is made by connecting the outermost points of an individual leaf. And then I look at the difference between the area of the convex hull and the area of the true leaf and divide by the larger area of the convex hull. So I basically get the proportion of white space in this second shape here, the convex hull image. And you can see that there's a lot more white space in the convex hull of a lobed leaf than in the convex hull of a round leaf. So in terms of looking at geographic variation in leaf shape from our common garden environment, we found that there's a very strong trend where high elevation populations of Mimulus acineatus have the most highly lobed leaves. And I just want to remind people that these populations are grouped into 3 altitudinal transects that are geographically separated, so geographically independent. And we see the same altitudinal climb replicated in each of those 3 transects. So the highest elevation populations from each transect also have the most highly lobed leaves. And this suggests that this pattern is due to selection rather than just gene flow among.
Sorry to interrupt. We're stuck on the leaf shape slide.
Okay.
Slide 10.
Just exciting. This one? Okay. What about now?
Yeah, it's moving now.
Okay. So what I said about this slide is that there is a positive cline where you see more highly lobed leaves in high elevation populations across the species range. And again, just to remind you, there are 3 independent altitudinal climbs within that range that I collected from, and you see this same pattern of high elevation populations having highly lobed leaves in each climb. We also see a pattern with latitude, where high latitude populations have the most highly lobed leaves as well. So, you know, high elevation and high latitude populations of Mimulus acineatus have the most highly lobed leaves. We see this in a common garden, so this is a genetic difference. And this suggests that potentially something about high latitude and high elevation populations has caused selection for increased leaf lobing, potentially cold, since that's an environmental factor that high elevation and high latitude have in common. So that's kind of the conclusions, that we see this pattern with high elevation and high latitude Mimulus laciniatus populations having highly lobed leaves. This elevation pattern holds across replicated altitudinal climes, and this suggests that these lobed leaves are adaptive in cold environments. And again, the physiological data of leaf temperature showing that lobed leaves stay warmer than round leaves may back us up with that idea that they're adaptive in colder environments. Okay, so what about the phenotypic plasticity patterns? So these patterns are kind of in the same direction as population variation. So meaning that there are more lobed leaves in longer days, so the same genotype produces more highly lobed leaves in long days, and high elevation populations, which have more lobed leaves in a common environment, end up flowering under long days because they flower later in the spring than short elevation or low elevation populations. So what we're doing now in my lab is we're planting these same inbred lines in multiple environments. So in wet and dry environments, in short and long days, in hot and cold, to look at the variation in plasticity across the entire geographic range. And we're measuring leaf shape using the convex hull, and we're examining variation in reaction norms. So in this, the reaction norm is the link between the phenotypes in different environments, right? And you can see that reaction norms vary based on genotype in this graph. So we're looking at variation in those reaction norms, but now for 180 genotypes rather than just a handful in that preliminary study I showed you data from. Okay, so now actually getting to the genomics, the theme of this working group. And all the genomic work for this project is ongoing, so hopefully I can come back in the future and present to you the results. But so we've generated whole genome resequencing data for 180 genotypes across the species range from our original grow out looking at leaf shape. And we've generated this at 5x coverage for each individual. And we're currently aligning this data to the Mimulus cutatus genome using the GATK pipeline. And that work is being done by my laboratory technician, Natalie Gonzalez, who is fantastic. And then our next step, once we have all that data aligned, will be to do a genome-wide association study for population level variation in leaf shape, but also for variation in those reaction norms, so variation in leaf shape plasticity. And we'll be able to ask, are the same loci involved in population level variation in this adaptive leaf shape trait as are involved in plasticity in that same trait? So does leaf shape plasticity have the same genetic basis as leaf shape, population leaf shape variation? So that will be really cool to be able to look at. And then beyond that, we want to know whether this variation in plasticity and geographic variation in leaf shape at the population level are either of those adaptive. So we have a suggestion that selection is operating on the population level with our repeated parallel climbs, but we'd really like to test that explicitly using our genomic data. So we're planning to do a genomic QST-FST to look at selection at population level and plastic variation in leaf shape. And then follow that up in the future with reciprocal transplants between high and low elevation sites using hybrids between high and low elevation genotypes that differ in leaf shape and leaf shape plasticity. So I just wanted to end briefly that my lab is looking for grad students and postdocs at Tulane, and I just got a grant this week, so I have funding, which is very exciting, and so contact me if anyone's interested. And then I have so many people to thank, my lab and collaborators and assistants and funding over the years. And you all for listening and bearing with me through these various PowerPoint related fiascos. So thanks.
Thank you, Katie. Does anybody have questions for Katie? You can type it in the chat, or you can just type a question mark. I can call your name and you can unmute yourself and ask your question. Okay, if not, I want to thank Katie again for the great talk. Our next speaker is Jose Maldonado. Jose is a PhD student at the University of Texas at Arlington, and the title of his talk is Genomics of Parthenogenetic Lizards.
Oh, present now. Hello, everyone. Thank you for being here. Like she said, my name is Jose. I'm a graduate student, and I'll be talking about some of the parthenogenetic lizards we work with and some of the big evolutionary questions we're trying to answer in our lab. So we know that sex is pervasive throughout nature, that even bacteria have some way of exchanging DNA between them. But when you start taking a critical look at sex, we notice that sexual populations suffer a demographic disadvantage compared to asexual populations, in which an asexual population will exponentially grow faster. And in just a few generations, asexuals should outcompete and drive sexuals to extinction. Now, this occurs because asexuals, everyone in the population will contribute to the next generation, but only half of the individuals within sexual populations contribute to the next generation. And this is famously known as the twofold cost of sex. So here it seems that sex is the evolutionary dead end, and that asexuality should be the dominant form of reproduction. But we don't see that in nature. It's the opposite, meaning that sex must confer some large enough advantages that it outweighs all of its cost. Yet identifying and determining these advantages has been somewhat difficult. Now, there's been multiple models that have been suggested to resolve this problem. And one of the models that has lots of promising is known as the mutation recombination model, that is surrounded around the efficacy of natural selection. And I'll use a visual to walk you through this model. So you have your original genotype. A new harmful mutation arises. Now within asexuals, the genome is completely linked, which reduces the effective population size of the linked genes, which weakens selection and allows for drift to heavily influence the genome and the trajectory of these harmful mutations, which they most likely will reach fixation. And after several generations, you're gonna have a genome full of mutations, which will cause genomic decay, a reduction in fitness, and eventually the asexual population should go extinct. Now, the opposite happens when you have a sexual population in which sex increases the effective population size of these genes, which strengthens selection. And then selection should be able to come in and select for these favorable alleles, but against the deleterious ones. And thus, only sex is able to create a new genome that has fewer mutations than the previous generation. Now, while there is a magnitude of theoretical support for this model, the empirical data is lacking, with like fewer than 10 studies have been published investigating this model within asexual lineages. And of course, what's been hindering studying this model was the cost of genomic sequencing. But now that we can cheaply sequence genomes and multiple of them, we can now really investigate the mutation recombination model, specifically in non-model organisms, which is perfect because the only true obligate asexuals that have clonal reproduction are found within squamates. Many of these asexual squamates have a hybrid origin between two distinct sexual species. And this gives us an opportunity to look into the asexual genome and understand the consequences of asexuality and make comparisons between the two groups. My lab specifically works with whiptail lizards in the genus Spidicillus, and we catch these fellows by hand and ladies. Both the asexuals and the sexuals are in sympatric with each other. So we can kind of assume that they have similar selection pressures. And because of this, we can make direct comparisons between the mutation accumulation rate in asexuals and sexuals. And the way we did this is by sequencing 83 whole mitochondrial genomes using a novel method, 47 asexuals and 36 sexuals with multiple species within the genus. We use all 13 protein coding genes, and we use DNDS between the two groups to quantify mutation accumulation. Now, we use DNDS, this ratio, to quantify mutation accumulation because we know when a non-synonymous mutation occurs, it's going to be slightly harmful because it changes protein function and structure. So if selection is weakened within the asexual lineages, we should see a quickly buildup of these type of mutations, but we shouldn't see them in the sexuals. And while a traditional DNDS of greater than 1 would imply positive selection, here would imply mutation accumulation. But given that, I wouldn't expect to find a high DNDS of greater than 1 within the asexual group, but the DNDS should be higher than whatever the DNDS of the sexual group is. Now, the mitochondrial genome is a perfect marker to study this because it's rapidly evolving. And while it does lack recombination, segregation still occurs between the mitochondrial genome and the nuclear genome within sexual populations. But when you transition to asexuality, you're going to abolish segregation, which should weaken selection's ability to act, which should allow for mutations to accumulate within this organelle genome. And the way we sequence our genome was a novel method that our lab developed, where we took our DNA extractions, we purified away the nuclear genome, leaving only behind the mitochondrial one. We then took a PCR way free to reduce introducing errors, and we exponentially amplified the mitochondrial genome. And then we did a standard library prep. Once we got our data back, we filtered them, cleaned them, and we were pretty stringent. We assembled them, we annotated our genomes, we aligned each gene, and then we concatenated our alignment so that we could make a large contig. We made a highly supported phylogenetic tree that you can see right here. Red branches are the asexual, white ones are the sexual ones. And the first thing we did was a simple model of a 1 ratio for the complete tree. And we got a DNDS of 0.443. Then we did the next model, which is a 2 ratio, where we compared the asexual branches versus the sexual ones. And clearly here you can see the asexuals have an elevated DNDS, about 2 times higher, signifying that the asexual lineages are accumulating these harmful mutations. And of course, we compared our 2 ratio model to the 1 ratio model, and you can see it's a better fit. We also did a 2 ratio model between the internal and terminal branches of our tree for all individuals. And we expected to see a higher DNDS within the terminal branches. And this is because there's a lag between new mutations occurring and selection acting to remove them. And of course, once again, we compare them to our models, and we see that, of course, it makes sense that the terminal does have this 2 ratio terminal versus its internal one is a better fit than the 1 ratio one. Lastly, we did a 4 ratio model where we allowed the internal and terminal branches for both asexual and sexual lineages to have different DNDSs. And in both cases, you can see that the terminal and internal branches of asexuals have an elevated DNDS compared to their counterparts. And then we compare this 4 ratio to the other models, and you can see that even the 4 ratio model is a better fit than the previous ones. So what is our data telling us? It's telling us that sex has to play playing a role in removing harmful mutations, that when you transition to asexuality, it's followed by the mutation accumulation. And here we're the first to observe an elevated DNDS within the asexual whip cells, providing the first link that transition to asexuality in vertebrates will also lead to mutation accumulation. And a future goal of our lab is to see if we see the similar pattern within the nuclear genome of the asexual whip cells. And really quickly, I want to talk about a paper that me and my co-authors just published in the annual review of Ecology, Evolution, Systematics, where we argue that we should be using, leveraging asexual vertebrates to answer the problems in evolutionary biology of why sex. And with that, I want to thank my collaborators, my lab, all here in this photo that helped me collect samples, my funding sources, the NSF GRFP, SSE, and the UTA Phi Sigma chapter. And with that, I'll take any questions.
Thank you so much, Jose.
Yes.
All right, questions for Jose? Are you typing a question? Matt has a question.
So if I understand it right, those earlier branching lineages in your phylogeny, did those ones accumulate more mutations over time versus the ones that branch nearer to the tips of the tree?
I'll go back to the tree real quick. Can you still see my screen?
Yep.
Mm-hmm.
Okay, so you're asking
asexual populations.
Yeah, so overall, we are seeing that the asexual lineages on the tree are accumulating more mutations when we compare them to the sexual ones.
Okay, next, Patricia has a question.
I was wondering, how did you, you said you developed a novel method to get only the mitochondrial genomes.
Yes.
Could you elaborate on that?
Sure. So going back to here, we actually found an enzyme that only digests away linear DNA, but because it chops away at the 5' and 3' ends, and because mitochondria is circularized, it doesn't have a 5' or 3' to clamp onto and kind of eat away and break away those bonds. And what we do is just do a digestive reaction to only target the linear genome, but it leaves intact the mitochondrial one. And we actually have a way of testing it that I didn't talk about to actually test that the mitochondrial genome is still intact and the nuclear genome is gone by doing simple PCRs. We PCR a nuclear marker and a mitochondrial marker, and you actually get a band for the mitochondrial marker you amplified, but no band for the nuclear gene you try to amplify, meaning that the nuclear genome is gone.
Thanks a lot. That's cool.
Yeah, thanks. Just a follow-up question on that. Have you tried that on museum specimens, or were these all fresh specimens?
This was all fresh specimens. I do, a previous graduate student in our lab, I think did work with museum specimens. And while in each genome that I sequenced, I would always get back the full 16 kb genome, he was actually able to recover just a few genes, which was good enough for what he was doing.
Cool, thank you. We have one more question from Rachel. Were there differences in overall nucleotide diversity between asexual and sexual populations?
I haven't looked at that yet, so I haven't had time to investigate that. But that's something we'll be looking forward to, and specifically more in the nuclear genome.
Great, thanks, Jose. There's comments on how good your talk was. But okay, our next speaker is Dr. Allison Shultz. Allison is a curator at the Natural History Museum of LA County. The title of her talk is Genomic Responses in the House Finch to a Novel Pathogen.
All right, thanks. It's great to be here and talking to all of you virtually. So I'm going to click over to my presentation now. I'm just going to kind of first introduce my thoughts here. So as an evolutionary biologist, one of my goals is to really try and understand both the patterns of biodiversity and the processes that produce them. And one of the ways that I do that is by studying evolution across different timescales. So for example, looking at interspecific patterns across big groups of individuals versus, getting into population genomics and even time series data. And I found that a great system to do that with is with host-pathogen coevolution. So here we've got, our virus and our bird. And so it comes as no surprise to any of us, given the current situation, that pathogens are consistently evolving to be better at infecting their hosts, and hosts are—
Sorry, Allison, we're not seeing your slides yet.
Oh, you're not seeing my slides. One moment. Okay, can you see now? Okay, sorry everybody. So here's my time series, but back to pathogens are consistently evolving to be better at infecting hosts, and hosts are consistently evolving to be better at fighting off these pathogens. And so they make a great system for studying across these evolutionary timescales, because you have, consistent across many different species, these relationships with many different pathogens, and you can actually study both sides of the equation. So far, in 2019, I published a big study in birds at the interspecific level, where we found that pathogens appeared to be the most consistent selective pressure across birds as a whole. We see increased selective pressure in immune genes, for example, and many actually of the same genes in birds and mammals being under natural selection. And many of those are viral interacting genes, which is really interesting. But today I have time to tell you about a study on the other side of the equation, which is time series. So really trying to look over very, very rapid timescales and actually observe the process of how evolution is happening. And the system I use for this is the House Finch. So the House Finch, which should be familiar to many of you, is a great system for studying ecology and evolution. It's a very common bird. Males have red on their crowns and their breasts. Females are kind of brown and streaky. And they occur on both the western and the eastern half of North America, although they're only native to the western North America. They were introduced to the east in about 1940. That's a whole nother story. So here's their range. But what's really interesting is in 1994, a chicken-associated disease called Mycoplasma gallisepticum jumped from chickens to house finches. And in house finches, it gave them conjunctivitis. In poultry and other birds, it's often a respiratory disease. And you can imagine that if you're a small bird and your eyes get swelled shut and you can't really see, your survival probabilities are not very high. Within 2 to 3 years, it traveled throughout the entire eastern population, and we saw 60% declines in some areas, so major population impacts. Just to put that in perspective with coronavirus, here we're seeing like 1% death and 60%. That is pretty crazy. It took about 10 years to get to the western population, which is the native population, and there it spread slower with a lower prevalence, but it's really picked up steam, especially lately. Just using iNaturalist, I've seen quite a few cases even this year, which is bad for house finches, but exciting for other reasons. And so, I'm not the first person to study this system. There are a number of researchers, especially at Cornell, and my PhD advisor, Scott Edwards, and his collaborators have studied this system and have shown experimentally that there's evidence for resistance and tolerance in the eastern population. And that means a population that had been evolving with the disease for some time. So, for example, they took individuals from Alabama that had seen the disease for about 10 years and individuals from Arizona that had never yet been exposed to the disease, or the population itself hadn't, infected them, measured their I-score, measured how much of the pathogen was present in their system, and found that the Alabama individuals both had greater resistance, so they had a lower amount of mycoplasma in their system, and also they were more tolerant. So given the same amount of mycoplasma, they had lower eye swelling scores and lower kind of overall pathogenesis. And so the question is, how did that resistance evolve? That's the one that I'm really interested in answering. Was it one gene? Was it via a number of soft sweeps? Was it one hard sweep that swept through the population? There are many different possibilities. And so kind of ideally to answer this question, one would be able to sample individuals before mycoplasma, and I'm referring MG, Mycoplasma gallisepticum, in both the populations, and then after you would also be able to measure to look at these same populations. And so while I don't have a time machine personally, it'll come to no surprise to any of you there that actually museums are a great window into the past, and we can use those as time machines to get an idea of what populations were like in a previous time period in a given place. And so I sampled 16 house finches per population per time period for a total of 216 house finch genomes. So here's my pre-mycoplasma, then 10 years after, and then in the East, I actually got 20 years after just because enough time had kind of passed by. I also sequenced a number of purple finches and Cass's finches, which are the outgroups to house finches, and more divergence finches, so I could get kind of accurate population genetic statistics. And so here's what my sampling ended up looking like. And so the two questions I'm going to address in this talk are, first of all, did gene flow from a resistant population lead to broader resistance and tolerance? So for example, did maybe a mutation arise and then sweep through the entire population, in which case we would expect to see spatially geographic variation before and after the disease? And if not, then how did house finches evolve resistance and tolerance? So here's a principal components analysis of the genomic data. And you can see that PC1 separates first the eastern and the western house finches, which doesn't come as a surprise, given that the eastern populations were introduced and they encountered a founder effect. PC2 actually separates the eastern populations, spatially. And if we actually lay that out, it actually separates them out by the distance from where they were first introduced in Long Island, New York, which is kind of a cool pattern to see. But one important thing is there's actually no temporal separation. So that suggests that there wasn't a lot of kind of major population migrations or gene flow that might be accounting for this evolution of resistance to the disease. So what are the other possibilities? Well, maybe we'll see a selective sweep or a change in allele frequency at a single locus. So just an example. So if we have a population of, say, green and bluebirds, but there's the evolution of resistance, which turns into a yellow bird. This might sweep through the population very quickly and also take along with it the variance on either side due to linkage disequilibrium. And so what would that look like if we were comparing the genomes in different time periods? If we were looking at how allele frequencies might change, well, we would see, some amount of variation and then probably a big spike if that occurred at one location in the genome, or maybe a couple if it occurred at several different locations in the genome. So another possibility is polygenic selection. And so this is if they occurred at multiple places in the genome, and probably those spikes wouldn't be quite as large because there wouldn't be quite as strong allele frequency changes. So what do we see with our data? I have genomic position on the x-axis and then the minus log 10 p-value on the y-axis. And so a higher value here means that there's a greater allele frequency change through time. So if I look at just eastern before mycoplasma versus eastern after mycoplasma, we don't really see that big spike or even a series of smaller spikes, at least nothing that we can detect. We have the power to detect with our data. And this is true for Western as well. So, this is, of course, not what you want to see when you're looking at your data. It's like, darn, that's not that easy story. And house finches overall have little differentiation through time. So, if we look at FST, this is a distribution for the before and 10 years after mycoplasma in the east, before and 20 years after mycoplasma in the east, and before and 10 years after mycoplasma in the west. And you can see it's perhaps not surprising that FST is very low, since this is, just a few generations of change in the same geographic localities. However, one thing that I mentioned when I was talking about sweeps is that it actually, not only do we see allele frequency change at a single locus, but there are differences at loci that are attached to them. So there are a number of other statistics that we can draw upon that might give us more power to detect how and where resistance has occurred, or how and where selection has occurred. So increased divergence is just one of those signals. But we also expect to see increased linkage disequilibrium around that locus because that single locus has swept to fixation or has become increased in allele frequency. We expect there to be differences into G-MIS-D, whether it's greater or lower, kind of depends on the type of sweep. So a soft sweep, you would expect G-MIS-D to be increased. In a hard sweep, you would expect it to be decreased. And you would also expect to see reduced genetic diversity around these regions as well. And so by looking at these different summary statistics and how these are occurring within and around genes, we might actually be able to pick apart some of these more subtle signals of selection. And so that's what I did. I calculated for each gene in the House Finch genome the average value for these different summary statistics, as well as 10 kb on either side, because I wanted to account for regulatory variation as well. I then got the basically outliers in these different comparisons through time and looked at the intersection of, for example, genes that had increased linkage disequilibrium, increased divergence, and decreased genetic diversity. And here's, we actually do get some genes that pop out. And so here's our Eastern before and 10 generations after, Eastern before and 20 generations after, and then Western before and 10 generations after. And so, we actually do see selection not just at one locus, but at a number of different loci, which suggests that it most likely is polygenic selection and also soft sweeps that are occurring, which is allowing these populations to evolve resistance and tolerance. Now I'm just going to kind of zoom in on one locus here. This is the locus BBX, which is actually a transcription factor. It was the most divergent of all of the different comparisons that we did. And just give you a sense of what this actually looks like when comparing our 3 different time periods in the East. So here we've got, genomic position. Here's where BBX is in the genome. This is FST. So this lightest purple is Eastern before and 20 years after. This darkish purple is before and 10 years after. So you can see it's only really very divergent in the later time period. But we actually see increased linkage to equilibrium in both before and 10 years before it has lower linkage to equilibrium, and then we have increased linkage to equilibrium in 10 years after and 20 years after. And linkage actually is tends to evolve before divergence does. So, this actually is probably part of the overall process. We also see reduced genetic diversity in this latest time period, and we see reduced T-G-MIS-D as well. And so this is, I'm kind of still ongoingly analyzing these data, but I think some of the questions that we can answer are that there is no evidence of spatial structuring through time. So there's no evidence that resistance evolved once and then spread widely throughout the population, and that house finches evolved resistance and tolerance through polygenic selection. And interestingly, there doesn't seem to be any parallel evolution in Eastern and Western populations. There are different sets of genes that have changed in allele frequency between the two. So with that, I'll thank my many collaborators, museum collections, funding sources, and I'll be happy to take any questions that you all might have.
Thanks, Allison, for the awesome talk. Anybody have questions for Allison?
I'll leave it here.
Raina asks, has the pathogen evolved?
Good question, Raina. The pathogen has evolved. So the pathogen actually has evolved to become more virulent in both places. It was actually a less virulent strain that jumped from east to west, and it since is evolving in both places. And so I personally am not studying the mycoplasma right now, but there are others that are working on this, and they have some whole genomes of the mycoplasma as well. So that's a whole nother really interesting story.
All right, I'd like to thank Allison again. That was a great talk.
Right.
Our next speaker is Dr. Durrell Kapan. You can probably tell from his outfit that Durrell is a colleague of mine at the Cal Academy. He's a senior research fellow here, and he's going to talk about an R program that he developed with his colleague, Bastian Pfeiffer, to estimate introgression.
Hi, everyone. Can you hear me?
Yes.
Okay, let me just get to it. Thanks a lot, Athena. And I just really want to quickly say, support your natural history museums, go museums. They do great things, and the Cal Academy is one of them. So I'm super happy to be part of this Genomic Social Hour, which, thank you, Athena, has been keeping us all connected and amazingly moving forward in this difficult time. So here we go. I'm going to go for it. Let's see. Of course, I didn't do it right. Hang on one more time. There we go.
We don't see it yet, Durrell.
You should see it now. There we go. Okay, so all of this work is the result of a collaboration with a person that I've never met in person named Bastian Pfeiffer, and he's in Austria at the Medical University of Graz, and wouldn't be possible without him. And it's also due in large part to the amazing open source community around the R software platform. Let me get straight to it. All the information in this talk is in this methodology article that you can get open access at BMC Bioinformatics. Easy to find because you can search for my name or Bastian's name. So what are my questions? I'm super excited about the origin and maintenance of biological diversity. And we know that biological diversity is organized at many levels, from between species to within species and even within individuals in a single family. Today, I'm going to tell a story about how trying to get a handle on the source of this genetic variation that creates these amazing colors and patterns in the heliconius butterflies led me to think a little bit deeper than I'm used to, and without Bastian's help, probably a lot deeper than I'm comfortable with, about how to measure when genes don't obey the traditional evolutionary routes through the species trees, so to speak. I put up air quotes, that when genes actually jump between species. In other words, when hybridization occurs and genes move from one species or taxon to another, in other words, introgression. So the search for introgression was made incredibly powerfully easier when we started to be able to have whole genome scale data. And it's often the case in science when a new method or a new microscope or telescope is built, we see further and we have to come up with new ideas and new methods to try to understand these new patterns that sometimes don't make a lot of sense given the old sort of mental structures we're using to understand the world. And one of the coolest methods that came out in the rise of the genomics era in the 2000s was this idea of counting SNPs on a tree which is polarized, a 4-taxon tree. In other words, we have an outgroup on the right, this example from our 2012 genome paper in Nature would be the sylvaniform or orange butterfly. And then if we see a mutation arise that marks a difference on the first two, in this case, branches to the left of that outgroup, we can use that to try to figure out whether or not those two basically branches or species or races in this case shared genes. And by comparing that with another possible marker mutation, we could get a different pattern, which if it was seen in excess would support the other direction of introgression from between the first taxon and the third taxon. So these two patterns are known as ABBA and BABA, and Nick Patterson and others utilized this in an early paper in Science on Neanderthals, and basically led to a whole cottage industry for counting SNPs. What does that look like? We all probably familiar with this, at least we've read about it. We're looking at a ratio of these two patterns, these SNP patterns, across a region like a chromosome, and comparing it to the total of those SNP patterns. So basically what we're generating with Patterson's D is a difference divided by a sum. Now this kind of ratio statistic can be powerful in that it quickly summarizes both the direction. If ABBA is higher than BABA, then this statistic will be positive. If BABA is higher than ABBA, then it'll be negative, as well as the magnitude of this sharing. And under the hypothesis that these two changes are random, then you should have that be 0. And so a significant difference from 0 will be something of interest. So great, let's just use it. In the genome paper, one of the key questions was, where do the color patterns come from when two species that are not closely related start to share warning color patterns? And here we can see the Patterson's D statistic for every chromosome, the 21 chromosomes in Heliconius melpomene and its close relative Heliconius timorata. And if you look at the number 15, and it's hard to quite see, but should be number 17, the location of both the genes that are for the lack of melanin on the hind wing that creates this little yellow bar, or the forewing dark red patch, show up as being highly differentiated for Patterson's D. And you can see that in this case, it's a ABBA, ABBA excess, and what we're saying is that both Melpomene amaryllis and Timaretta are sharing genes. So we're done, right? We don't need to do anything. Well, as we look a little closer at these statistics, we realize that some of them are somewhat problematic. We've seen this with FST. As you get smaller and smaller gene regions, when you're doing a gene scan, you're able to sort of get extreme values due to low diversity. So some problems with Patterson's D was it wasn't originally designed to actually go at the gene or base pair level for a scan. And as a ratio, we found that it's not directly proportional to the signal of introgression. And because this low genetic diversity problem occurs, we can get false positives, and it has a very high variance. And this is related to this FST problem, which was discussed in the middle of the 2010s. This idea of genomic islands or areas of extreme FST differentiation also have similar issues with the fact that low diversity can create more island-like areas. Now, could those be real? Are those differentiating areas, or are those due to statistics? So this led me to a really kind of obvious thought. Introduces genetic distance, like duh, we all know that. So why would we care? And a hypothesis, past or recent hybridization will leave a signature in reduced DXY. Of course, that's obvious. So what can we do with that? And I thought long and hard and did some exploratory analysis and realized I needed somebody to help me. But basically, I wanted to inspect that sort of dumb idea. Can we use this obvious prediction to build a new way of looking at introgression that doesn't have this problem? So that's the whole point of today's talk, and it's kind of an advertorial or a non-tutorial. It's an advertisement. So we did a twofold thing. This is, and I won't be able to get into the, I might be able to get into this in the questions, but we wrote Patterson's D, and actually another statistic that I don't have time to get into, in terms of genetic distance. In other words, we had the enemy of genetic distance, and we wanted to figure out if we could use it as our friend. So the very top is Patterson's D on the right. It's that ratio of ABBA to BABA, ABBA minus BABA divided by the total. And through some magic that is called math, we were able to actually rewrite this in terms of DXYs, and this is the basic output. So you're just gonna have to trust me for a minute. It's all in the paper. But what was cool is that if we take the numerator, which is the good part of this, we essentially have something pretty similar. The distance between 1 and 3 minus the distance between 2 and 3 times these allele frequencies at the in the population 2 and the population 1 are sort of like a weighted amount of ABBA and BABA. The denominator does show you why you have problems when you have low genetic diversity in the system. So if we take the numerator, can we fix the denominator? So what Bastian kind of spent some time thinking was that if we added in the genetic distance between the in-group, the first and second taxon, and the rest of the tree as a ruler controlling diversity, we might be able to fix the denominator and have less of a problem with these extreme values. And so I'm jumping to the final thing, skipping all the math. Our new statistic, distance fraction, was born, and it's essentially much easier to understand if we use graphics. So without introgression, we have the 3 taxa of interest plus the outgroup. We get essentially 2 measurements we're trying to compare. The first one is the light blue line between 1 and 3, and the second one is the dark blue line between 2 and 3. And so in this case, the distance fraction is proportional to the difference between those 2 pathways on the tree divided by the total. But what's interesting is that both of those lines have this measuring stick in it, which is shown by this ruler between the node P12 and P123. So that's the sort of normalizing bit that went into the math. So when we don't have introgression, what we see is DF is approximately 0. And when we do have introgression on the right, you can basically say at some time a marker, which is this black dot, which is shared in the P3 branch, followed along with the introgression of some genomic region into P2 in this case. So when we look at D13, the difference we'll see no change in the length. But when we look at D23, we're just going to be missing, because there's no variation between P3 and P2 at that region. It'll be smaller. So in this case, the ratio will be greater, or not the ratio, but the fraction will be greater than 0. So essentially what we're looking at is pairwise distance comparisons. What does this look like in real world? First we tested this with simulated data. And here we're basically showing the same kind of coalescent process. The time for gene flow is TGF, the direction of gene flow, and we're going to be comparing the DF statistic in orange, Patterson's D in blue, and another statistic in the paper we don't have time to go into, which was an effort to fix this problem that came out in 2015 called FD. The actual prediction, the real fraction of introgression, the correct answer would be these red lines. And you can see for gene flow coming from 3 to 2, it's a pretty good fit for the orange ones. Whether or not the time to gene flow is quite a bit since the splitting of the third taxon from the second and first, or whether it's kind of at a star much more recently. Doesn't always work as well the other direction. This is one of the not so great things about all of these statistics. But one thing you should notice is that Patterson's D is highly biased. In both cases, it shows quite a strong increase for a small increase in in fraction of introgression. So this evaluation goes through lots more things in the paper, but we varied distance to ancestral population size, the time of gene flow, recombination rate. And for the first two, the distance fraction is as better or equal than this intermediate statistic, and it's much better than Patterson's D. It's overall superior for the time of gene flow. There's no change there's no bias depending on the time of gene flow. And the two alternative statistics to Patterson's D are approximately the same when you change recombination rate. So essentially we have a new statistic that has some good properties, still goes over 0 to 1. It works for small regions. It's designed for gene scans. Inside, how do you calculate this? This is built straight into R, the PopGenome package, which also does all your basic population genomics statistics. And Bastian built in the ability to smooth your data, to do false discovery corrections are available with R, and we can take advantage of R's jackknife and bootstrap methods. So one of the problems with this is that we don't always have a special case of 4 taxa where we can polarize our SNPs. And there are 3 and 2 taxon methods that do exist. But this is an area of investigation. How can we think about genetic distance and control for it? So there's a number of people working on this now, and I'm excited that we kind of figured this out pretty early on in the game. How does it work in practice? This is a bit of a confusing graphic, so let me go over it. If we use this Fontaine et al. Anopheles Gambier VCF, which you can download from the internet, we wanted to look at this interesting chromosome inversion along the 3LA chromosome. Their science paper examination of this is on the bottom. And they show Patterson's D being significantly negative in this purplish blue color. And it's basically across the inversion. And for comparison, I just redid this today and just say, hey, is it still working? Is this package great? Even a year later. And I was able to calculate the D statistic in gray and the distance fraction statistic in blue. And you can see right away that I'm able to get the same numbers. And the distance fraction isn't quite as large, which makes sense because we put in an extra amount of variation in the denominator. It's no longer a ratio, it's a proportion of the total variation. And the interesting thing here is we can put in the amount of the nucleotide diversity between the key taxa in that comparison. And you can see that even though the introgression across the inversion is fairly high, fairly consistently negative, the DXY isn't looking particularly different. By the way, that's multiplied by 10 just so you can see it. So it works. You can learn how to use this by going to CRAN, the Comprehensive R Archive Network. Look up PopGenome. Specifically, there's the URL. There's a bunch of vignettes and a reference manual. And the two things I'd like to mention, if you're using the paper as your guide, Bastian is in January 2020 updated everything on CRAN. So now all you have to do is say install packages PopGenome, and you want to load that package using the library command. It's amazing. You can do FST, McDonald-Kriepman, almost anything you can think of that's standard inside of R, and it's relatively rapid. All of that work I did to show you that last graph, I just reran it, and it took maybe like 3 minutes before this talk. So thank you very much for your attention. I'd like to thank people who commented on our work and inspired some of our efforts, as well as this discussion group and similar journal club in Austria. And I got to really hand it to Bastian. This is what it looked like. He's a Linux guy. When we talked, his camera was flipped, but he really is not a bat or a vampire. He's amazing guys, and continuing to build that package that can help us to do all the work we're interested in.